So I'd like to welcome everybody. Is this on? Doesn't matter. I'd like to welcome everybody to the second Storer lecture given by Simon Levin. And he needs even less introduction today, and I think he probably wants more time. So really pleased to have Simon again. His title's up there. Let's welcome him.
There, my microphone's on, right? Okay, no response. I figured it was off. All right, I want to talk to you today about evolutionary perspectives on ecosystem patterning. And first of all, a few words about why I think this is an important thing to do, why an evolutionary perspective is important. At least to me, I am concerned about the status of environmental systems. And when one worries about the loss of environmental features, one's usually thinking about macroscopic patterns that really characterize systems. Of course, we worry about species loss too, but we worry, there's a lot of emphasis now on ecosystem services, and this means dealing with macroscopic patterns and what makes them robust. And macroscopic patterns are, of course, the result of evolution, but it's a rather distant, in a certain sense, in that the evolution's operating at much lower levels of organization. So how do we make the connection between the evolutionary processes that's operating in these complex adaptive systems at quite low levels of organization and the fact that there are regularities that exist at the level of ecosystems and the biosphere? So the central question I want to address is how are ecosystems organized and what maintains the resiliency of these macroscopic patterns like nutrient cycling and other things I'll talk about today. How do we explain the regularities we see at the level of ecosystems in the biosphere and the homeostasis that maintains functioning? Lovelock proposed a view which I will not accept. Often what all I have to do is put up this slide and people come up to me afterwards and say, how can you believe that? So I don't. But Lovelock was a great scientist and is a great scientist, and his view is that the biota play a role in mediating and maintaining the physicochemical environment, with which I think we would all agree. That's why we're concerned about the loss of biodiversity. But it's the second half of the sentence that becomes worrisome. It is, again, kind of an anthropic principle that's at work, that they maintain it at just the right conditions for its survival. So there's a coevolution going on. That's why those biota are there, because those are the conditions that allow it to exist. So in the extreme form, the problem with Gaia is that the biosphere becomes a superorganism that's somehow been selected for its macroscopic properties. And that's not a point of view that a population biologist is very comfortable with. The problem is that Gaia describes macroscopic regularities, but evolution's operating at lower levels, not for the benefit of whole systems. As I said yesterday, ecosystems in the biosphere are what have come to be known as complex adaptive systems. That is, they're made up of individual agents, like in this orchestra that you see here, that interact locally and evolve. And the reason I put an orchestra instead of a genetic system is to make clear that when I talk about evolving here, I don't mean in the narrow evolutionary sense. It can be individuals adapting their behaviors to respond to other features in their environment. So based on the outcomes of their interactions, they change their behaviors, or their changes may be genetic. And from that, macroscopic patterns change as well. So patterns emerge then to a large extent, not entirely, from phenomena at much lower levels of organization, in particular at the level of individual agents, small spatial scales, and short temporal scales. And I want to understand how these phenomena then interact with things that are going on in broader scales. So my lecture will have three parts, not equal in length. The first, I will talk about some approaches to the evolutionary ecology of resource use, which I think is fundamental to understanding the organization of ecosystems. Talk a bit about selection for prudent resource use, and finish by saying a few words about the evolutionary ecology of ecosystems patterns in general. So to the first part, motivated by one regularity, a debated regularity. I think one of the questions that I won't have time to talk about today is the question of how do you identify invariance in systems? What are the things that characterize ecosystems, and how invariant are they? And if they're not, I think it's useful to think about these as invariants even if they're not invariant, because then we can at least look at them as indicators of particular types of systems. We can look across systems and see how these variables change and why. So marine systems show a remarkable constancy in the ratios of key elements, carbon, nitrogen, and phosphorus in particular, even though the absolute levels of these things may vary across the oceans. The ratios of them are relatively constant in the water column, but also if you look at the phytoplankton, the primary producers, or at the zooplankton, the consumers of these phytoplankton, you get the same sorts of ratios. This was an observation now 3/4 of a century ago by Redfield, who asked the question of, is this a bottom-up or top-down control, namely, to which I'll come back, or is it that the organisms are just responding to those numbers in the environment, and one has to look elsewhere for an explanation of those, or are the organisms somehow controlling their environment? And the ratios, depending on whether you look at the organisms or the environment, of, for example, of the nitrogen to phosphorus, are 16-to-1 or 15-to-1, roughly, and carbon to phosphorus about 106-to-1. And this Redfield, this is the observation Redfield's probably best known for, and people have argued ever since about the explanations. So this is what I'll talk about in the first part of the talk. And I will say, first of all, and this comes from a paper that we published in Nature. That's Chris Klausmeyer, who was my postdoc, Elena Lichman, who was still is Chris's wife, and Paul Falkowski's postdoc, and I and Tanguy Dufresne published in Nature a few years ago. I should say at the beginning, if I forget to make clear, if I ever say I really mean we. And if I say we, I usually mean they. So, so this is really work that Chris and Elena did in going to the literature and looking at 29 species of phytoplankton at their N to P ratios. And you can see that well, this is the Redfield ratio of about 16-to-1. But although the average is in some broad sense, and this is laboratory data, so it does, there's no way to weight these to find out what the true average is, but they bracket 16-to-1, but there's a lot of variation. So in addition to asking why is the ratio 16-to-1, we need to ask why is there so much spread, and why is there coexistence of all these types. To what extent are the ratios, as Redfield asked at the beginning, in the water column simply integrating what the biota do, or is it the other way around, that the evolutionary patterns in the biota are just reflective of what the environmental inputs are, or is the truth, as I believe, somewhere in between? So the approach that we took to this problem, and then I'll take to the other problems I'll talk about, is to separate timescales and to think about things on the ecological timescale and assume the evolutionary timescale is longer, and that works sometimes. It's not always going to be the case. So I will begin by developing a model of what's going on the ecological timescale, and then allow mutants, variants, to be introduced on longer timescales and ask, in the spirit of Maynard Smith, can they invade? Are there evolutionarily stable strategies to which the system converges? So this is the ecological model, and here we'll basically assume there's a single, to begin with, that there's a single type of phytoplankton species in the environment, and this is what's going on. This is the input to the water column. The organisms take up nutrients and put them in storage, and then operate. It's like going to the store. They acquire these nutrients. Some of them they use right away, and some of them they don't. And then, based on what they have in storage, they build new biomass, And in building new biomass, they take into account the stoichiometry that they need. And we will consider organisms that have different stoichiometries to see, in some sense, what's best. So let me explain this in a little more detail. I don't want to go into all the mathematics of what's going on here, but I can put people onto the relevant papers if you're interested. So this is the water column where there's phosphorus and nitrogen. And P in and N in are the inputs that are coming from the external environment. And here we simply assume that the net uptake is a Fickian diffusion proportional to the difference between the inputs and what's in the water column. So this is the net input into the water column, and then things are taken up by the organisms and put in storage according to uptake functions that basically depend on proteins that govern the uptake. And for these, we usually use Michaelis-Menten types of uptake functions, but we've also looked at other forms. There's an uptake function for phosphorus and one for nitrogen. B is total biomass, so the uptake is proportional to biomass. And then things are put into storage. Now these terms, these M terms, are death terms or settling out. These are losses to the system. But the key is that, and I'll show you the specifics of these equations, the organism then has a growth function G, and phosphorus is taken out at rate alpha, nitrogen at rate beta, and that goes into making new biomass. So pictures are easier for some people. For me, the equations are easier to see what's going on. So I'll just translate what I just told you into equations. This is what's going on in the ambient environment. There's a net input into the water column, and then there's a loss due to uptake by the organisms. And then in terms of what's in storage, these are the input terms that you see here, and then there's a loss from storage into biomass at a rate that's proportional to mu, which is the growth rate, times, and this is the key function, and it involves two assumptions. In terms of growth, it's known as Droop's law. It's a fairly standard law in the phytoplankton literature. And the key here, which we are modifying now, is that your growth rate depends on the minimum of two quantities determining which nutrient is in shortest supply. So this is Liebig's law of the minimum. So that's what's going on with this growth function here. And then that determines what happens into biomass. So this is the set of equations that turn out to be, for a mathematician, interesting anyway, because there's a kind of discontinuity here, at least in the first derivative, that makes analysis difficult but not impossible. So we can show, and for mathematicians, there are formal proofs that depend on the theory of monotone systems of this, that the system goes to a globally stable equilibrium as it runs out of some nutrient. Well, what that means is that the system reaches an equilibrium, and it reaches the same equilibrium no matter where it starts, and the outcome does not depend on initial conditions. It always ends up the same place. So the picture looks something like this. This is phosphorus, this is nitrogen here, the system starts here, and it runs down as it uses up both nutrients, but it eventually stops when one or the other runs out. Which one runs out depends on the input parameters and on the those parameters there, as well as upon the phenotypic parameters, whether the organism needs nitrogen or phosphorus more. So now, that's the ecological time scale. It's a straightforward analysis. On the evolutionary time scale, the approach we take, which was really Chris Klausmeyer's insight, is to think about that organisms have to allocate carbon. By the way, this model has to be modified if you want to think about it, say, for trees or something, where they're putting a lot into structural biomass. Organisms have to allocate carbon to either to proteins or to ribosomes. They can put them into proteins that are good for uptake of nitrogen or phosphorus, or they can put them into ribosomes, which they need for growth. But these proteins and ribosomes have different N-to-P ratios. Each one has a characteristic stoichiometry, meaning C to N to P ratio. The allocation pattern, the way you decide to use the carbon you have in proteins versus ribosomes will therefore determine the overall stoichiometry of the organisms, the alphas and betas, and thus indirectly how much is being allocated to uptake versus growth. So if we do this evolutionary analysis, let the system come, what we do is we let the system come to an equilibrium and then ask whether another type can invade, and we do this over and over again. What we really do is we look for evolutionarily stable strategies, or technically continuously stable strategies, which means evolutionarily stable strategies that are evolutionarily attracting. And the first thing we find out is that the system always evolves to co-limitation, so that it's not limited by nitrogen or phosphorus alone. In other words, it ends up down here on this corner. Now, intuitively, this should be clear that the reason this works is because if the system's limited by nitrogen and there's a variant that comes in that makes more use of the available phosphorus, then it's not limited, it can grow, and so it replaces that type. And the only time you can get to an uninvadable strategy is if you have co-limitation. If you're up here, you start to drift down here. If you're over here, you start to drift over here. And the system evolves to co-limitation. And not only does this fit with post hoc intuition, but my colleague Francois Morel, who advised us on parts of this, sort of intuited this and is a believer. This is sort of faith-based science on his part that the systems must evolve towards co-limitation. The second thing is if you look at the evolutionarily stable allocation to growth versus uptake, what you find is represented by this graph here, where the color indicates how much of the allocation to growth there is. And what you're supposed to notice on this graph is this is an almost uniform color until you get down to extreme phosphorus limitation. Namely, the stoichiometry, the evolutionarily stable allocation pattern, doesn't have much to do with the external conditions. The partial answer here to Redfield's question, which is that, and I will repeat this on a slide in a moment, that the evolutionarily stable allocation is being determined by the stoichiometry of proteins and ribosomes more than by the external environmental conditions. So the organismal evolution is not just responding to the environment, but it is, in a sense, in terms of what it excretes and dead bodies, determining the environmental conditions. Unfortunately, we were pleased when we got this result, but we were getting ratios of about 45 to 1. Remember, the right answer is 16 to 1. So the evolutionarily stable allocation to uptake is larger than what we would observe with Redfield ratios. So we thought about this and we decided that the reason was the equilibrium approach that we were taking. Whereas if you, for those who are familiar with these sorts of systems, let me come back to that second half of that sentence in a minute. So the question then is, why is this different than the observed ratios? And we still haven't addressed the question of why do we get coexistence, because this model always gives us the same equilibrium, always come to that same point. So for those who are familiar with this literature, you will remember that Hutchinson's paradox of the plankton. Hutchinson, one of the greatest, if not the greatest ecologist of the last century, wondered why so many phytoplankton species were able to coexist both in marine and freshwater systems, even though they were competing for a small number of resources. How did you have, given that you had the theory of competitive exclusion, which basically says the number of species ought to look like the number of resources they're competing for, how is it you have so many more? And his argument was it's because of the non-equilibrium conditions in the ocean that you're getting changing in conditions that first favor one type and then favor another type. Disturbance is one way that happens. And so we reasoned that we really had to think about the non-equilibrium approach of the system. And in a fluctuating environment, as Hutchinson said, different species are going to be favored at different times, and in particular, following a disturbance, then those that could grow fast are going to have an advantage, essentially the early successional types. So we went back to the same model, and instead of looking for an evolutionarily stable type, we simply said, what is the type that would maximize the growth rate? And that type is a type that has to make a larger investment in ribosomes, higher P usage because that's what ribosomes have, and in fact we get an answer of about 8-to-1 if we do that. So if the equilibrium approach gets us up about 45-to-1, the maximizing the growth rate gets us about 8-to-1. So very crudely, we are bracketing Redfield. We're showing for the most part the range that we were getting, although there was one extreme type that was way out here. And what our hypothesis is now, and what we're working towards, is that we need to be looking at a spatiotemporal model in which we're imposing disturbances. And this fits with the growth rate hypothesis of Sterner and Elser, which says that as disturbance creates opportunities in particular regions, it will favor those that have low N to P ratios who will come in early and be displaced by those with higher N to P ratios. We'll get a successional sequence, and our hypothesis is if we look at a spatiotemporal model with localized disturbances, we'll get something that looks a lot like what's really being observed in these systems. We'll certainly get this range from favoring these and then ultimately these as the system reaches local equilibrium. So just to summarize that, by allowing for environmental fluctuations, we think we can approximate the Redfield ratios well, and also explain coexistence. We've extended this in various ways. In fact, the first paper we published on this it dealt with light limitation. It was it's the same paper that I mentioned in Nature of a couple of years ago. And we also looked at what happens if you say, well, let's suppose just the system is light limited or N limited or P limited, and that changes things slightly. You get different ratios out here, but not a whole lot different for what the optimum would be. And as I just mentioned, we're looking now at spatiotemporal fluctuations and mixing, and and that's related to successional patterns. This is closely related to work that Mick Follows and Penny Chisholm have been doing in trying to explain the coexistence of different ecotypes, largely bacteria, in their data by a spatiotemporal model in which they select possible species out of a spectrum of possibilities and try to simulate ecosystem evolution. They're not doing anything analytical, but we're looking for ways to converge these points of view. Now, I talked only about marine system. I just want to say a few words about other systems. My colleague Lars Hedin has been looking at such phenomena in terrestrial systems. It's not easy to make comparisons because you don't have a water column in the terrestrial systems. You have to measure N to P ratios in outflow from systems and things of that sort. But Lars and his group have been trying to estimate the N to P ratios in terrestrial systems. And in some systems, like coniferous forests, he's getting things that look a lot like marine systems. This is just to give you some idea, however, of the range and work that Lars and Megan McGrady, his postdoc, have published. You can get ratios these are the marine systems. These are global forests. You get higher N to P ratios. So these N to P ratios vary across systems. And Lars and his group have started to modify our models to look at these, but they have to be different because of the role of carbon in those systems. So I'm not going to say anything more about that. People who study terrestrial systems observed that even in systems that are nitrogen limited, there don't seem to be as many nitrogen fixers as you would expect, and Therefore, or related to that is the fact that you get reduced uptake in these stable terrestrial environments. Why? So Duncan Mengi, who is a joint student with me and Lars, has been looking to extend these models to think about the evolution of nitrogen fixation. And this is very much work in progress. It's his thesis work, actually. So I'll just say a little bit about that. He considers that, again, the system has two resources. Could be nitrogen and phosphorus, but nitrogen and some other resource. And he modifies our model. This looks a lot like the model I showed you before. It's the ecological model. But this is the biomass of the species. This is nitrogen, and this is the other resource. And basically, this modifies our model by allowing the organism to fix nitrogen and ultimately asking evolutionarily what's the optimal strategy, given that there's some cost to nitrogen uptake. And ultimately, this has to be put into a spatial framework as well. So again, this is embedded in an evolutionary model, and I'm not going to report on the details of this except to say that sometimes that co-limitation is not necessarily the outcome here. In the simplest case, you do get co-limitation, again, if otherwise the system would be nitrogen limited, but under more complicated uptake dynamics, if the uptake rate of the other resource increases with fixation, you don't necessarily end up with co-limitation. You can get a nitrogen fixer that's still limited by the other resource. But I don't want to scoop his results. These results, however, should be published within the year. The other thing we're trying to do, which is of technical interest to people, I think, in the ecosystems literature, is to get away from the assumption that's standard in the ecological literature of Liebig's law of the minimum, that is, that there's this sharp transition from limitation by nitrogen to limitation by phosphorus, for example. A similar problem is approached in the economics literature where, for example, capital and labor might be the limiting factors to growth. And there one recognizes these trade-offs, but also that there's some degree of substitutability. And as long as there's some substitutability, the function doesn't have this sharp discontinuity. It has a more continuous shape, which on the one hand makes it easier to analyze because you don't have to worry about these complexities. On the other hand, the whole notion of co-limitation sort of disappears because everything's always co-limited. It's just a question of the degree to which it's limited by one thing or another. Now, it may be that under broad conditions, these are non-substitutable resources, nitrogen and phosphorus. But in other situations, we think that this continuous arrow-solo type function is well worth being introduced into this literature. So let me return to the question of what might account for reduced uptake in stable terrestrial environments. And this is, I'll talk to you about work of another graduate student, Eduardo Zea, together with a hydrologist at Princeton, Ignacio Rodriguez Iturbe. And this deals with the evolution of water use strategies in terrestrial systems. Now, nutrient limitation is, in this sense, limitation of water because water is the single limiting resources in many systems. And yet, again, we have a great diversity of types that vary in their morphology or physiology. Again, we build a basic ecological model. Plants have to balance processes, assimilation and evapotranspiration, by modifying their stomatal dynamics. And the standard approach in the literature is to optimize uptake rates or something of that sort. But we avoid optimization approaches because it loses the frequency-dependent aspects of what's going on. Resource use is in a global commons, and so again, we're going to need game theoretic approaches and spatially explicit models. So we start from an ecological model again, in this case a physiological model that takes all of these processes into account, and then we look at the evolutionary dynamics by introducing variant types that have different transpiration rates. And the one result that I want to show you here is represented in this graph. What we do is we modify the spatial regime in the following way. At one extreme, every plant has its own water table underneath of it, which it doesn't share with anything else. At the other extreme, all plants are drawing from the same water table, a global commons situation. And then we consider the whole spectrum in between, namely different levels of mixing of the, of the water table that belongs to different plants. And the basic result we find is that the evolutionarily stable transpiration rate increases almost linearly with the mixing rate, so that if the mixing rate is low, then you have an optimum transpiration rate down here on this scale of about 1 unit, which goes way up to about 8 units if everything is well mixed. And again, this fits with some of the things I was talking about yesterday. And Jonathan, it fits with some of the things we talked about this morning, namely the more the mixing, the more the sharing of the resource, the less your prudence will reward you later, and therefore the more selection for overconsumption now, because why leave it there if it's going to be used by other individuals? So this works in fisheries and it works for plants as well. So that's all I want to say about prudent resource use. Let me turn then to the last part of the talk, which is to think more generally about extending these approaches to think about other ecosystem patterns. I focused on stoichiometry as the example worked out in detail because there's so much interest in that and so much data that's around to look at that problem. But stoichiometry is just one set of the robust patterns that we see in ecosystems. Sheldon's particle spectrum, which describes the power law decrease in the size of particles ranging from the largest organisms down to the microorganisms, is a remarkable constant in marine systems. And these are various plots that are typical, that show you what was first recognized by Sheldon, that is, you have this power law relationship if you look at biomass size spectra. And looked at it in the right way, the total biomass in every bin is basically the same. It falls off in the right units with about a -1 slope. So why is this? So together with Zach Powell and Charlie Stock, who's our joint postdoc, Zach, many of you know because he used to be here, we've been building bottom-up models to try to understand in the same way why we get these macroscopic patterns so regularly emerging, even though the interactions are at the much lower scales of organization. How can we understand the emergence of these regularities from evolutionary forces? I think that's the great challenge. The patterns that that Lovelock, for example, is interested in are remarkable and characterize systems. They are repeatable with some variation, but they're not being determined at the macroscopic level. Why do we see these emergent patterns? Spatial distributions also show characteristic patterns. This is work that will be familiar to many of you. Some of you have worked on these sorts of phenomena. If you look along gradients in the oceans at the distributions of characteristics, these are Dave Mackes's data, then temperature here, this is along a spatial gradient. Temperature is almost constant with a little bit of bumpiness to it. Phytoplankton, this is actually chlorophyll, probably fluorescence, is a little bumpier, but zooplankton are much bumpier. So you can look at these data in a different way. These are not the same data. These are Weber and El Sayed's data. This is a spectral plot of data of that sort, temperature, fluorescence, and in this case, krill distributions from the southern oceans. And what you see is there's a fall off. This is a power spectrum. So this is the broad scale here, and these are the finer scales here. And what you see is there's a fall off in in the patchiness as you, I'm sorry, in the variant, the power that exists at different cycles as you get to the broader scales. Temperature and fluorescence don't look a lot different. The Krill distribution looks like temperature and fluorescence maybe at the broad scales, but not at the fine scales. The explanation being here that there are large water movements and that the temperature spectrum reflects that, and that are moving the phytoplankton around and moving the zooplankton around at the broad scales, but then zooplankton are active swimmers, and so they're showing patchiness on finer scales as well. What accounts for these regularities? So these sorts of spatial and temporal patterning, I'll remind you, also hark back to the stoichiometry and help to resolve some of those patterns of coexistence because we have all of this patchiness. So how can we understand the emergence of these sorts of regularities from evolutionary forces that are operating at lower levels of organization? Can we develop rules that will let us scale from the small to the large, from the microscopic to the macroscopic? Even bacteria aggregate through quorum sensing and signaling for collective benefits. And this is an area that's just opening up. People who are interested in applying the insights from ecology and evolutionary biology to microbial systems that have usually been looked at species by species in the past. Can we develop evolutionary approaches to understand this sorts of behavior, the signaling, quorum sensing, why they arise, how collective behavior emerges, and the patterns of aggregation? More generally, how is it that individuals, as I discussed yesterday, coordinate their motions to produce schools and flocks? How do we describe it ecologically, and how do we describe it evolutionarily? So there's been a lot of work over the last 25 to 30 years on the description of the aggregation pattern. And in particular, and this is especially work of my student Danny Grunbaum, in proceeding from microscopic laws that are Lagrangian in that they follow individual organisms, mass times accelerations, the sum of the forces acting on an organism. Some of those forces come from the fluid dynamics, some from organisms simply following chemical gradients, and some from them orienting towards each other. Can you, and the answer is yes, derive macroscopic descriptions by developing a statistical mechanics of these ensembles and then proceeding to Eulerian descriptions similar to the Navier-Stokes equations, but reflecting how organisms relate to each other. So there's a large literature on that, but mostly it treats all the organisms as the same and doesn't address the evolutionary questions. But in this approach, if organisms are responding, and I introduced this yesterday, both to environmental information and to each other, how does it trade these off? How does it balance the information it gathers for itself against the information it gets by observing others or signaling others? Now, there are evolutionary reasons that for aggregation. Basically, it may relate to collective foraging, in other circumstances to protection against predation. In other circumstances, aggregation is to facilitate reproduction. Whatever the reasons, one can begin to look at evolutionary models, give rewards in terms of fitness to particular strategies, put types in competition in the same way I described for the stoichiometry, and ask what are the optimal strategies. How can we understand these behaviors in evolutionary models? For example, if organisms like these krill are searching on some resource landscape that looks like this, if everybody just follows their own information, some individuals will find their way up here, some will find their way up here, it would be better to share information because in collective foraging you have a better chance of finding the optimal peak. On the other hand, if everybody just followed each other, they'd end up in one clump. It wouldn't have anything to do with the peak. So from the collective point of view, the best is some intermediate strategy. And from an evolutionary point of view, that's not the right question anyway, because every organism is going to be maximizing its own payoff function. And so how does selection trade this off? So we've begun to look at that within the context largely, and I'll just repeat a little bit of what I was talking about yesterday, by looking at models in which individuals have their own rules and their own weighting functions, and we modify those. You may remember that I talked about this updating function, which is a balance between following others and following your own information with this weighting function, omega. Omega is the property of the phenotype here, and we allow that to evolve in order to find out what's the best strategy. For example, if I pulse nutrients at different points in the system and measure fitness in terms of how much resource an individual gets per unit time, I can introduce new types and examine what the winning strategies are. So those are the sorts of things that we are looking at now. So let me conclude. In environmental protection and conservation biology, a central problem is how to understand, how to characterize what makes ecosystems robust. That involves some of the things I talked about yesterday, characterizing ecosystem services, and understanding what makes those services robust. All of that means we have to be working across scales. We have to move from cells to organisms, from organisms to collectives, and ultimately up to the whole ecosystem. And to ask questions like, are these properties of ecosystems robust? So that's a measurement question. The literature on the Redfield ratios is coming under a lot of criticism and analysis recently because people who are looking at it carefully are finding a lot more variation than has previously been assumed. Does the answer to this depend on scale? Do you find particular regions of the ocean that have different stoichiometries? In other words, and can you look at this genetically and say there are different genotypes or different sections of DNA that are associated with different regions of the ocean? Can we relate them to the characteristics of that region of the ocean? As Jonathan Eisen and I and a group are hoping to look at, are there particular regions of the ocean that select for novelty, or other regions that select for collective behavior? How can nothing of this sort has been done at this macroscopic level. And how does the robustness of the macroscopic properties depend upon what's going on at the finer scales? And ultimately, can we use an evolutionary approach to explain these macroscopic patterns by building agent-based models, developing rules like those Lagrangian to Eulerian rules to extrapolate across scales, and thereby relate the microscopic processes and the patterns and processes on the broad scale? How important are individual species? Do we have to pay attention to individual species, or can we think in terms of functional groups such as these nitrogen fixers? Ultimately, I think there are great opportunities, especially in marine systems, by marrying theory and empirical work to elucidate, first of all, what the patterns are with regard to these macroscopic measures, and explanations of why these measures might vary across systems, such as stoichiometry, or even within systems, and develop, ultimately, predictive models for how these respond to changing environments. And I think that only by providing this bridge between the microscopic and the macroscopic are we going to be able to develop a predictive theory. So thanks very much. Jonathan.
So I have a question about the Redfield ratios. So you mainly talked about the possibility that organisms could adjust their relative allocation to growth versus uptake in particular situations.
Yeah, and they might do that through phenotypic plasticity, or they might do that, or that might represent genetic change within the population.
So on the evolutionary level, they could also adjust the relative need for the uptake and growth systems to use particular nutrients. How much flexibility is there in that? Have you looked at that side of things? I mean, can you change how much phosphorus is needed for those systems in any robust way?
Do you mean they can change it by basically filling a different niche in the system?
No, using different amino acids or proteins or
I don't know.
amounts of RNA in the ribosome to therefore make better use of a resource-limited environment over an evolutionary chain?
Well, that's certainly theoretically possible. I don't know the degree to which that's known, what's done, but that's certainly something that would be possible to do. Yeah, Rick.
Is there any evidence from experimental evolutionary studies of, say, phytoplankton, which have short generation times, that in fact populations can and do evolve to a local optimum, not just, genetically evolve to a local optimum under selection when different ratios of limiting resources are manipulated?
Yeah, I'm probably, is Sharon Lawler here? Because she would be probably would know about that better than I. My understanding is there's evidence, at least from lakes, that goes on. Steve Carpenter has talked to me about this. I don't know the data on that, and I suspect that that this has been done in laboratory conditions as well. But obviously, that's an important question to ask. I just don't know the answer. I don't see any other hands, so maybe nobody else knows the answer to that either. Yeah, Alan.
So most of the examples you talked about were sort of without really strong species interactions other than through sort of a single resource.
Yep.
And would you care to speculate on sort of what adding, for example, much more, say, top-down effects and avoiding and dealing with predators, how is that going to change?
Yeah. So let me answer that question in two ways. First of all, we have done some stuff of that sort. My postdoc, Josh Weitz, has been very interested in that, especially in the role of viruses in mediating diversity in these systems. It's one thing to ask, suppose you have a predator or virus operating on these systems as a fixed agent, what's it going to do? But the reason, and we've made some progress on that. I thought about even talking about that. But the reason so far looking at these sorts of systems is, as you know, once you start looking at the parameters of species interactions, there's so many parameters. And if you start, for example, with the classical equations like the Lycko-Volterra equations, and say that the competition coefficient is evolving, well, that's the competition coefficient is a property of both species that are interacting. So I'm uneasy with that because it doesn't tie to a measurable change in genotype or phenotype. It's an emergent property of the interaction. So the nice thing about looking at competition, exploitation competition, is that here you can talk about it in terms of the properties of organisms. Ultimately, one has to extend to interference mechanisms, but I'm persuaded that if you're going to do that, it's really best to do that by understanding the mechanisms that give rise to that interaction as properties of the organisms, and that's a much more complicated thing to do. But I agree, it has to be done. Yeah, Don.
But in answer to Alan's question, the German phytoplankton and zooplankton people have done 25 years of work showing that these competition coefficients are resolved at the resource level, the minimum resource level at which the population can maintain stable population. And so the winners in these zooplankton contests are the ones that can maintain equilibrium at the lowest resource level. And
yeah, that's also the Tillman sort of.
Yeah, and then they drive the other species out.
Yeah, but that argument, which I agree, is based. I mean, that's basically the approach that we used to do the analysis on the Klausmeier system. But that really is based on models of exploitation competition, with organisms taking up things from the resource. What I was stepping back from is extending that. And that works very well. But when you have interference competition and poisoning and those sorts of things, then things become more complicated. And so I'm perfectly comfortable with the resource limitation sort of approach that the species that win out are those that can drive the system, can persist at the lowest level of the resource. That's exactly the same approach that's used in dealing with epidemiological systems, where the virus that can drive the susceptible pool to the lowest level is the type that wins out. But it's these more complicated situations that involve not interaction through the resource, but interaction by direct contact that are more problematical to measure.
So I hope this isn't repeating some of the questions that have been said.
I hope it is. It makes it easier.
I'm wondering, you talk about looking at stoichiometry in a particular system, the nutrients going in and then the nutrients coming out. But what about interactions between trophic levels and looking at the stoichiometric relationships there?
Yeah, we haven't done that yet. It's obviously the thing to do. I mean, we've looked a little bit at what it was implied in Alan's question, which is not what you said, namely, suppose that there's some loss in the system due to predation. How does that, or to the activity of viruses, how does that affect things? But we haven't looked at the zooplankton in terms of their utilization patterns. We don't want to do that till we get this straight, but obviously that's a next step. Looks like they're all played out, Alan.